Hi everyone, welcome to the course of structural biology. We are continuing towards introduction and I have talked about history, evolution of biology. Today we will talk about biological macromolecules. We talked about light microscope and resolving power. We have already seen the contribution of physics based technique to biology had begun with the development of the microscope. It marked the beginning of the studies of cell structure, the structural cell biology, what it is known for. And after that, improvements in microscopy have facilitated even more revelation of the cellular and subcellular structures. Nevertheless, visible light dependent microscopy has its own limitation of the resolving power. As the resolution was inversely proportional to the wavelength of the observing light, the microscope had reached the theoretical limit of its resolving power. The problem could be overcome by the introduction of electron microscope, where visible light waves are replaced by electron waves, which are bent and focused by electromagnetic lenses, hence achieving much more resolution. But when we are talking about biological macromolecules, even the normal electron microscopes are not good enough. So, we will discuss about that, about the instrumentation, but let us take a look when starting from an organism coming to organ, coming to tissues, cell, organelles, you will see that the tissues are ranging within 1 millimeter to 100 micrometer. The cells which we are talking continuously are ranging from around 100 micrometer to 10 micrometer. Organelles are within the range of 1 micrometer to 100 nanometer. The biological macromolecules comes in between 10 nanometer to 0.1 nanometer, and you could easily see that light microscopy could not reach that area. So, how to study them is what the structural biology techniques talks about. But again, before going into them, we will try to look at what are those biological macromolecules, what are their basic properties and all these things. So, towards our journey to the macromolecules of life, we have certain questions in our mind. What about the macromolecules of life? Who are they? What are their functions? How they grouped, how they chemically linked? How are they composed of? How we could read them, which is extremely critical. We are talking about structure, but to get to the structure, we have to read the sequences. So, how we could read a DNA, how we could read an RNA, how we could read a protein molecule, how we could read lipid, carbohydrate, which are easier, which are not possible, or which have technical limitations, all this is our discussion. And at the end, definitely, what about their structure? And if you look at, we will discuss, but why you don't see carbohydrate or lipid structures to be discussed. So, coming to the introduction of biological macromolecules, for survival and growth, living organisms require nutrient, but they are not being able to synthesize them. Animals obtain nutrient by consuming food, while plants pull nutrient from soil. Many critical nutrients with polymeric nature are called biological macromolecules. The term macromolecule was first coined in 1920s by Nobel laureate Hermann Staudinger. Staudinger was also the first to propose that many large biological molecules are built by covalently linking smaller biological molecules together. He have introduced the concept of monomer to polymer formation of the biologically relevant molecules. Biological macromolecules play critical role in cell structure and function. There are four major classes of biological macromolecules which we are going to discuss. One, carbohydrate, protein, fat and nucleic acid. Beside nucleic acid, which comes in a different way, carbohydrate, protein, fat are part of our daily diet. Let us start with carbohydrates. Carbohydrates serve as fuel and building material of the body. Carbohydrate includes sugars and the polymers of sugars. The simplest carbohydrates are monosaccharide or single sugars. Carbohydrate macromolecules are polysaccharides, polymers composed of many sugar building blocks, and that polymerization can be straight chain or it could be branch chain, and that is one of the difficult thing to study carbohydrate. If you look at these bondings, they could be formed in many ways. First of all, the monomers join to form polymer in units connected by single bond. What is the problem with connectivity of a single bond? If you start thinking, you take a pen which is accessible from your table and you just try to rotate the pen, you will see that the pen is easily rotatable. Now, if you take a second pen, I do not have, but you take a second pen and you try to rotate them, you see that you cannot rotate them freely. So, when two units are connected by a single bond, they are freely rotatable, which means they could have generate each and every conformer. So, it is difficult to predict which conformer would be stable. Whereas, if there is a double or triple bond formation, there are restrictions, and these restrictions we will discuss in detail while discussing protein macromolecule. Monosaccharides have molecular formulas that are usually multipliers of CH2O, carbohydrate, carbon and water. Glucose C6H12O6, that means this is CH2O whole 6 is the most common monosaccharide. Monosaccharides are classified by two ways. One, with the location of the carbonyl group. If the carbonyl group is at the top like this, then they are keto. If it is in between, then they are aldose. So, they are ketose and aldose. So, you could see here glucose and fructose, you will see the difference, ribose and fructose also. Also with the number of carbons in the carbon skeleton. So, if you see here 1, 2, 3, so they are called triose. So, glyceraldehyde is a triose 1, 2, 3. If it is 5, they are called pentose. Ribose, deoxyribose are pentose. If it is 6, they are called hexose. Glucose, fructose are hexose. Coming to fat, fats are constructed from two types of smaller molecules, one glycerol, two fatty acids. Glycerol is a 3-carbon alcohol with a hydroxyl group attached to each carbon. So, this is glycerol, 3 hydroxyl groups are there. A fatty acid consists of a carboxyl group attached to a long carbon skeleton. A long carbon skeleton with a C double bond OH. So, if you look at capric acid, lauric acid, oleic acid, linoleic acid, they are all example of fatty acids. Now, this fatty acid and glycerol, they fuse and form a ester linkage to form fat. Fats are of two types, saturated fat and unsaturated fat. While butter, which you use regularly in home, is an example of saturated fat, olive oil is a good example of unsaturated fat. Now, look at the structures, and there is a very interesting thing to observe. This is the structural formula of saturated fat molecule. You see them, when it is saturated, this goes straight. And this is the space filling model. These are representing stearic acid. Here you see both saturated and unsaturated. You will see the double bond, and because of the presence of the double bond, it is unsaturated, but more interestingly you will see a nick there, kink or nick. Because of the kink present here, what happened, when they are only straight like this, they actually set there in the membrane and they reduce the dynamicity of the membrane. Whereas, when it is having this bend, they are more dynamic. As a result, from your practical experience, you will find that butter is solidified in most of the condition, while olive oil is liquid in most of the temperatures, even if you keep them in refrigerator. So, this cis double bond is very critical to define the nature of the fats. Now, here another very interesting thing you have to keep in mind, these are called monosaturated. So, for monosaturation it come and then it have the cis double bond and then it have a bend. But in case of polyunsaturation, so polyunsaturation, what will happen? It will have a bend, but it will again have bend. So, ultimately it would be getting the straight saturated like structure. So, saturated are bad for dynamicity, monounsaturated are good, polyunsaturation again giving the same scenario like saturation. Coming to phospholipids, in a phospholipid, two fatty acid and a phosphate group are attached to glycerol. If you see, this is glycerol, CH2OH, CH2OH and CH2OH, two of them are occupied by fatty acids and the third one is having the phosphate group. The two fatty acid tails are hydrophobic, as we know before, but the phosphate group and its attachment form a hydrophilic head. Because it is phosphate, it is not always that it would form negative charge. In the presence of choline, which is showing here, it shows positive charge in the hydrophilic head, and in that way phospholipid could show negative or positive charge, creating the environment negative or positive. Steroids are lipids characterized by a carbon skeleton consisting of 4 fused rings. If you see, 1 ring, 2 ring, 3 ring and 4 ring. So, 4 ring fused together. The best example of steroids which are present in our body is cholesterol. It's an important steroid, is a component in animal cell membranes. Although cholesterol is essential in animals, high levels in the body may contribute to cardiovascular disease. Coming to nucleic acids are polymers called polynucleotide. Each polynucleotide is made of monomers called nucleotides. Each nucleotide consists of a nitrogenous base, a pentose sugar and one or more phosphate group. Depending on them, it is called mono-di-tri. The portion of the nucleotide without the phosphate group is called the nucleoside. If you look at this animation, it would make it more clear. So, there are three components: the sugar, nitrogenous base and the phosphate group. The first one is the sugar, the second one is the nitrogenous base and third one is the phosphate group. Now, they only differ in the nitrogenous base. In DNA, you get adenine, guanine, cytosine and thymine. Instead of thymine, in case of RNA, you will get the uracil. Coming to protein, proteins include a diversity of structures resulting in a wide range of function. Proteins account for more than 50% of the dry mass of the most cells, and protein function includes structural support, storage, transport, cellular communication, movement and defense against foreign substances. Protein is what we are going to study throughout. So, we will talk about protein in more detail. So, I am just touching it in this case. Proteins function. Proteins are generally of two types. One is called functional protein. They are also called enzymes. So, enzymes are used for selective acceleration of chemical reaction. Very interesting. If you think your body is a chemical factory, but your body have to maintain physiological conditions, physiological temperature. So, if you want to perform a reaction between A and B, you go to your chemistry lab, you put A and B, you could put heat, temperature, pressure and other conditions so that the reaction proceed in the forward direction. You cannot do that, right? You cannot, take the things and sit down on the oven and burn yourself. Don't do that. So, you have to accelerate by other way, and the other way is provided you through enzyme. There are proteins which are involved in defense, which protect against disease. For example, antibodies inactivate and help destroy viruses and bacteria. There are storage proteins, for example, function of storage of amino acid. For example, casein is a protein of milk, is the major source of amino acid for baby mammals. Plants have storage protein in their seeds. Ovalbumin is the protein of egg white used as an amino acid source for the developing embryo. Transport proteins, hemoglobin, people have studied myoglobin, hemoglobin in more details. They are the proteins which are crystallized at the initial stage and we got their structure, and they have developed as a model for many things like hemoglobin, how it forms its oligomers, the tetramers and all. So, hemoglobin is the iron containing protein of vertebrate blood, transport oxygen from the lungs to other part of the body. Other protein transport molecule across cell membranes. In case of protein, the building blocks of proteins are amino acids. Here they have a amino group, they have carboxylic group, they have one hydrogen and they have one R. R means X. There are 20 different R's, so 20 different amino acids are there. Now, if you think about, proteins are developed by permutation and combination of these 20 different amino acids. So, it gives a lot of diversity in character. We will study about those in much detail. If you look at the basic, and for the part of this course, one thing I will request to read and memorize, that is the chemistry of the protein. I will talk in details, but to understand the beauty of how biology is working, it is extremely essential to remember in your head where the chemistries are taking part, and for that you have to know all the chemistry of the amino acids. So, there are basic amino acids like histidine, arginine, lysine. There are acidic amino acids, aspartate and glutamate. There are hydrophilic amino acids like glutamine, uh, asparagine, cysteine, glycine, serine, tyrosine, threonine, and others are more like hydrophobic. Again, we will talk about them in much details. Proteins form beautiful three-dimensional structures. What is the target of this course? To study and understand. And protein being the functional molecule of the body, you have to understand the three-dimensional structures they are developing. If you look at individual proteins, contains very different three-dimensional structures, and if somebody will provide you the structure, you start guessing about the function. That is the beauty of the three-dimensional structure. Chemistry meet biology. This is the portion which is very, very interesting and critical. If you see the periodic table, only 6 elements, hydrogen, carbon, nitrogen, oxygen, phosphorus and sulfur, they develop the entire biology. And if you listen to me, I would say mostly the chemistry is developed by hydrogen, carbon, nitrogen and oxygen. So, it is very, very critical to know about their chemistry. Starting with carbon, because carbon develop the base of any biological molecule, be it the protein, be it carbohydrate, be it the macromolecules, cell, everywhere. It is the carbon whose neutrality is what we are depending our life for. If you look at carbon, carbon have 6 electrons. So, by electronic configuration is 1s2, 2s2, 2p2. So, carbon have in its ground state 4 electrons, which are divided like this in the 2s and there are 2 electrons here. In the excited state, this electron jumps here and this process makes 4 electrons, 4 unpaired electrons, and not being S and P, they are all now sp3. So, they create 4 sp3 hybrid orbitals. What is hybridization? There are many complex chemical theory about that. If you ask me, I consider hybridization like making hodgepodge, which means khichdi in our language. So, when you prepare hodgepodge, what you do, you take pulse and you take rice. You mix pulse and rice in a ratio. When the hodgepodge is prepared, you get a mixture. You could realize that there is pulse, there is rice, but you cannot realize which one, or you cannot separate them with their individual identity. Hybridization is also like that. There is involvement of s orbital, there is involvement of p orbital, but from the sp3 hybrid orbital you cannot separate s and p. Again, more interestingly, if you have a little experience of cooking, you could tell me that with the increasing ratio of pulse, the taste would be enhanced. Similarly, from sp3, where S is 25%, P is 75%, to sp2, where it is 3366, to sp, where it is 50-50, the properties would be significantly altered. But for biological purpose, it is good enough for us to discuss sp3 hybridized carbon, nitrogen and oxygen. When we will go to the protein part, we will do a further look on the peptide bond, and I will talk about the sp2 hybridized peptide bond. Okay. So, let us start with this carbon. So, this carbon is now having 4 sp3 hybridized orbital, which help the carbon to form a tetrahedral structure with the angles 109 degree 28 minutes. If you take a look, this would be a carbon look like when it form covalent bond with 4 hydrogens, it will form methane. So, what will happen to the other two, the nitrogen and oxygen? Let us take a look. Similar to carbon, nitrogen have 7 electrons. So, in the ground state it have 2s and 3, 2px, 2py, 2pz. In the excited state, they will form again 4 sp3 hybridized orbitals, but one of them would contain a paired electron. So, what would be the structure look like? This is the structure of a typical hydride of nitrogen, which is ammonia. But now you have a question probably in mind. You told when there are 4 sp3 hybridized orbitals, they are going to form tetrahedral. Why this nitrogen is forming 3 bonds? Because when you want to understand that, you have to take a more interesting take. I have taken that model. So, this is a carbon model, but I have introduced a lone pair here. So, what would come here? The nitrogen would have 3 orbitals with single electrons with one lone pair. Okay. The single electrons would form covalent bond with hydrogen, and that will give NH3. Can this lone pair form bond with hydrogen? Yes, but in that case it would not be considered as covalent bond. This would form a new type of bond which is called coordinate bond. In the coordinate bond, these two electrons would be shared between hydrogen and nitrogen, and this will give nitrogen ability to form positive charge. So, you can see now this is the way the nitrogen would be looking. It have 3 hydrogens, but it could form a extra bond which is called coordinate bond. Okay. Is there anything extra? Yes. The lone pair actually repel the bonds, which is known as, there is a theory which is called Vesper valence shell electron pair repulsion theory. According to Vesper, if there exists a lone pair, it would push the or repel the bonded angles. So, from 109 degree 28 minute in carbon, here it would be reduced to 107 degree approximate. What will happen to oxygen? Oxygen is having 8 electron. So, in the ground state, it would have 2s and 4p. In the excited state, again it would form 4sp3, but there would be 2 lone pairs. So, how it affect? So, basically you know hydride of oxygen is water, right? But as I told you, I made a carbon equivalent model of oxygen where you could see that there are 2 lone pairs and 2 hydrogens. Now, what will happen? You already know, according to Vesper, the repulsion would be more, and this now from 109 degree 28 minute, the angle reduced to 107 degree, it would be further reduced to 104 degree 50 minutes. Do you know what is the effect of this? That is where life exists. Because of this angle, now oxygen perfectly form hydrogen bonds. Because of these hydrogen bonds, they could be able to change their physical state, exist as a liquid. If you look in the periodic table going back, you will see that in the upper part of the periodic table, most of the hydrides are gaseous. Only exception is oxygen because of its ability to influence by Vesper getting the angle of 104 degree 50 minutes, able to make hydrogen bonds, and that is the reason. Now, if you think you have studied in your class 10, 12 level, where you have studied in physics a lot of special properties of water, like water form ice which float in the ice. So, the liquid is having kind of denser, because of this network they have a lot of vacant space, and you will go back, there are many further properties which are influenced because of that. Before finishing this part, I would also include, if you look at the periodic table, you will see that hydrogen, helium, lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine, neon, sodium, magnesium, aluminum, silicon, phosphorus, sulfur, chlorine, argon. Do not be afraid, I am not going to write the entire periodic table. What I am trying to show is, see, nitrogen and phosphorus share a lot of properties. So, you could identify the properties of phosphate in terms of the property of nitrogen, but the difference is definitely going down the periodic table, it reduces the electronegativity, right, and the size increase. Similar to that in oxygen and sulfur. One interesting thing, just to end this, you will see carbon and silicon here. Now, if you go to the market, you will see a lot of plastic replacement are this silicon, because silicon poses similar property like carbon. This happens. There are other complex properties which I am not going to discuss in this class. So, because of this only presence of their 4 electron which are distributed equally, carbon explains the fact why it is so neutral. And as I told, you could similarly argue about silicon, which is correct, only thing is the presence of vacant d orbital in case of silicon do not allow it to become carbon. So, carbon can form vast number and variety of compounds. It is vital to organic and life processes. It can form 4 covalent bonds, often seen in covalent interaction in hydrophobic regions. Nitrogen, critical for many biological molecules. Ammonia, often the starting compound for many nitrogen compounds, can form 3 covalent bonds, often seen in covalent or hydrogen bonded interaction. Oxygen, very reactive, combines with most elements component of many organic compounds, can form 2 covalent bonds. Hydrogen binds to carbon, nitrogen, oxygen atom, not very reactive, can form 1 covalent bond, participate in hydrogen bonds, which is extremely critical in biology especially. Phosphorus, very reactive, key element in energy currency in biology, which is ATP, can form 3 or 5 covalent bond. Sulfur, similar to oxygen, found in 2 essential amino acids, which are cysteine and methionine, can form 2 covalent bonds. Now, coming to a very interesting part, I call them 3 secrets of covalent bonds, and it is very interesting to look at in this basic part, which we will be repeating in a huge way when we are considering the macromolecule. First thing is configuration. What is configuration? Configuration is the spatial arrangement of atom in a molecule. For example, a carbon atom with 4 single bond bonded to 4 different atoms shown in this picture has different configuration. Changing configuration requires bond breakage. Configuration defines the molecule's chemical reactivity and type of bonding it participates in. Let us take a look. These are two same molecule. You will see that they have this green part, they have this blue part, they have this ash color bigger molecules and hydrogens. Now, if you change the position of one of the bonds, let us say you just change the position between them. Now, you see these are still same molecule, but you could never superimpose them as I could have done before. These are, first of all, when carbon is having 4 different groups like that, the carbon is called chiral. Which is chirality? I am coming later. Now, when you have chirality, you have dextro-levo 2 molecules with same elements but different configuration. That is where the configuration is important. Why configuration is so important? Think about, and I will give this example, this is very favorite to me. Think about, you are a guy. You are standing in a bus stop and you are waiting for bus. The bus arrived and the bus have 2 doors and you have equal chance to decide in which door you access. But suddenly when the bus stopped in the bus stop, you have seen an extremely beautiful girl in the driver's seat. That would change your mind, or that would fix your mind to choose the front door. That is called biasness. Similarly, in a protein, because of this chirality, we will talk about among the 20 amino acids I talked about, 19 of them are chiral, and because of them protein have a chiral environment. That chiral environment decide a reaction or a substrate to approach from which side. If you look at this, you will clearly see that if you want to put this carbon in a plane, you would be unable to put 2 of the atoms in that plane. Now, if you see, one atom is small like hydrogen and one atom is a big or a big group, the ligand would choose this direction. That is the importance of configuration. Coming to conformation. In conformation, if you see, you have to perform something which is called a rotation. So, if you have an amino acid like this, if you look at this structure is representing an amino acid which is called alanine. If you see, this is the carbon, here is the hydrogen, here is the carboxylic group, here is the amino group. So, amino acid, hydrogen, and the R group is methyl. So, it is alanine. Now, you could see the position if I rotate these, so from there all the other groups being same, the oxygens would be changing their position. This is called conformation. So, spatial arrangement of atoms in a molecule that can be obtained by rotation of the atoms about a single bond, no bond breakage required to change conformation. So, as I have shown you, you could see if you look at, there is rotation and it could go in this side, it could come in this side. So, that is called conformation. I have already explained chirality, but still if we have to understand, this is a molecule with 4 different groups connected to carbon. If instead of one of these I would put a hydrogen, then it would lose its chirality. So, chirality is the property of molecule that makes the molecule and its mirror image non-superimposable, which we have discussed earlier. If there are no chiral center in the molecule is not chiral. So, configuration, conformation and chirality are 3 secrets of covalent bonds which are utilized towards formation of these biological macromolecules and take very significant part in their property development. Why do the 3 secrets matter? Shape of biological molecule, in when we will go for folding we will explain in more detail. When we discuss stability in protein engineering we will talk about this in details. Recognition and function of biological molecules, I have talked about the biasness of chirality. So, binding, interaction, because of these 3 secrets: configuration, conformation and chirality. Coming to the building blocks, as we have already discussed, amino acids are building blocks of protein, nucleotides of nucleic acids are building blocks of DNA and RNA, monosaccharides are building blocks of carbohydrates, glycerol and fatty acids are building blocks of lipid or fat. So, how the biological macromolecules do their composition and show the function? In terms of property, protein use the atoms carbon, hydrogen, nitrogen, oxygen and sulfur. Protein get phosphorus through phosphomodification. Nucleic acid use carbon, hydrogen, oxygen, phosphorus and nitrogen. Carbohydrate use carbon, hydrogen, oxygen and nitrogen. Lipid use carbon, hydrogen and oxygen. The building blocks, as I told in the last slide, for protein it is amino acid, for nucleic acid it is nucleotide, for carbohydrate it is simple sugars and for lipid it is fatty acids and glycerol. Can they form polymer? If yes, what is the bond type? Protein, yes, it could form polymer. The bond type is peptide bond. Nucleic acid, yes, the bond type is phosphodiester bond. Carbohydrate, yes, glycosidic linkages of various type. Note the various type. The presence of various types of bonds makes it impossible to further study of carbohydrates. Carbohydrates are very important. You could see in all the edible foods, you get cereals and all, you get carbohydrates. But because of the presence of various glycosidic linkages, it is nearly impossible to model their structures. That is the problem. Lipids does not form polymers, they form various di-triglycerides. What are the intramolecular interactions? For protein, covalent bond, disulfide between the cysteines, hydrogen bonds, charge based, which is salt bridge, van der Waals interaction, base stacking and all. For nucleic acid, hydrogen bonding, stacking interactions. For carbohydrates, hydrogen bond. For lipid, mostly van der Waals, hydrophobic. Functional role in the cell, Proteins, as we discussed, work as enzymes, structural proteins, sensor, receptors, etc. Nucleic acid is a storage of genetic information. It also works as enzyme in the form of ribozymes. Carbohydrates, storage of energy, structural formation, recognitions, interactions. Lipids, membrane formations, signal, energy storage, small molecule like hormones. Now, we will make a shift towards a very critical direction. We have talked about the biological macromolecules, but it is very interesting to know, is it possible that we could read the biological macromolecules or not? If possible, how? What are the technologies? How easy to read them? And if we read one molecule, can we connect that to the other molecules? So, we have talked about the basic properties of the macromolecules. Now, we will shift ourselves to decode them. Decoding the biological macromolecules would be extremely interesting, because the first step from going to looking at a molecule to get the structure and understanding function, the first step is to read. It is like you want to write a novel and you don't know alphabets. So, our first target is to understand if we could decode the biological macromolecules, what would be the technologies? If yes, how easy those technologies are, how financially competent those technologies are. More importantly, how one technology could be connected, or reading one macromolecule, we could get to know about information about the other macromolecules or not. And I will finish here. I hope you enjoy this session. We have discussed about the biological macromolecules, their different properties and function, the chemistry behind them, and the composition and comparatives of them. Thank you very much for listening. I will be coming in the next class with decoding biological macromolecules. Thank you.